Okay, good morning. I'm doing a little test run this weekend to see if I can successfully do a video of one of the lessons that we did this week. And also I thought it might be an opportunity to go over autosomal linkage, which was something that we covered quite quickly. So I thought you might want to have another way to go over it. So first of all, see if you can remember what happens to the chromosomes during meiosis. Using this chromosome bank down here, see if you can remember where you would put the different pictures of the chromosomes in the different daughter cells. You can pause the video, have a think about it, and come back in a second. Okay, so hopefully you had a little go at that. And I've got an animation here ready just to recap on what happens during meiosis. So during meiosis, we first have the two chromosomes lined up here. Now let's say, for example, that this blue one here is the paternal chromosome and the red one is the maternal chromosome. So let's follow. So first of all, in meiosis 1, what happens is the two homologous chromosomes line up in what's called a bivalent in metaphase down the middle of the cell. So they pair up like this. And at this point, later on, we'll look at what can happen when we have an exchange of genetic material between the chromosomes. So now we move into anaphase. And the chromosomes separate. Sorry, here's the genetic exchange actually. And then anaphase is going to come up when the two chromosomes separate. There we go, anaphase, moving into telophase. And of course in meiosis, that's not the end of the process. There is a second division, which we call meiosis 2, where those chromatids in each chromosome are then separated. So we end up producing 4 different unique gametes with varied genes on them. Okay, so that's meiosis. So did you get the starter right? So this is what you would be looking for. So in the parent cell, you would have this chromosome and this chromosome. Crossing over would produce a little bit of an exchange. So you sort of have something like this. And then you would get, well, no, sorry, hang on a minute, it's not that one. It's this one and this one. And in the 4 daughter cells, you'd have 4 different chromosomes, each genetically distinct. Let's check that's right. Yeah, there we go. Okay, let's go back to the slideshow. So, first of all, let's recap what happens in normal dihybrid inheritance. Okay, so this is normal dihybrid inheritance here. So here we have 2 pea plants, and we're looking at the seed color. So we've got some peas which have round yellow peas, and we have some pea plants which give wrinkly green peas. So yellow color and round shape is dominant, and wrinkly and green color is recessive. Now, if we breed these 2 pure breeding strains together, we get what's called the F1 generation. So the F1 generation is a cross of the 2 pure breeding strains together. So the F1 generation, in this case, all of them are round and all of them are yellow. And that's because the dominant characteristic kind of takes control. Now, what happens if we breed the F1 with the F1? We call that self-fertilization. So if we allow those plants to self-fertilize and F1 crosses with F1, then we get this sort of classic dihybrid cross. And if you look at that and you sort of work out all the genetics, all the different possible combinations of gametes, you will eventually get a ratio of 9 to 3 to 3 to 1. So 9 of the sort of 2 dominant characteristics, 3 each of sort of 1 dominant and 1 recessive characteristic, and then just 1/16 will have the kind of recessive characteristic. So that's classic dihybrid inheritance. But what about linkage? Let's look at that. So linkage is something different. Linkage can cause ratios to be different. So in linkage, we've already said that the classic ratio that we expect is 9 to 3 to 1. But in linkage, sometimes we can have a different ratio. So Mendel also investigated pea plants, which were sort of purple and white flowers versus long and short pollen. And he was expecting his 9 to 3 to 1 ratio, but in the end, he actually got something different. Sorry, actually, it wasn't Mendel. It was Bateson and Punnett, two different scientists. So Bateson and Punnett did this experiment. Actually, I'm going to go back and edit this. Okay, start again. So this is linkage. Now, after Mendel, two geneticists called Bateson and Punnett performed a cross looking at the pollen length and also the colour of the flower of some pea plants. So they bred 427 pea plants. This is the F1 generation. So he had some F1 generation of purple flowers with long pollen. And then they bred bred lots of these F1 plants together. And they did it 427 times. And they would have expected a 9:3 to 3:1 ratio. So you can go ahead and work that out. You'd have to do 9/16 times this number to work out this. And you'd see what the expected results would be. If you'd like, you can pause the video and just work that out now. When they actually figured it out, however, the ratios were very different. The actual results were 296 to 19 to 27 to 85. And the reason for this is called linkage. So, linkage in pea plants. Linkage is where two genes are inherited on the same chromosome. So here's what was going on. In the pure breeding parent strains, these are the genotypes of the two parent strains. You've got big P, big L on one chromosome, and it's the same on the second chromosome. And in this pure breeding parent over here, we've got little p, little l on one chromosome, and it's the same on the second chromosome. So, what happened in the F1 generation? Well, in the F1 generation, inheriting one chromosome from this parent and one chromosome from this parent, we've got this genotype. So we would say the genotype is big P, big L, little l. But crucially, the P and the L, the big P and the big L, are held on one chromosome and the little p, little l are held on the other, which means we don't get as many different combinations because the chromosomes are not assorting independently, which is normally what happens when we produce the gametes from the F1 generation. So, therefore, in the F2 generation, we can only pass on either a big P, big L together or a little p, little l together. We can't pass on a big P and a little l or a little p and a big L because the genes are physically stuck on a chromosome. So this means we get a 3:1 ratio. That is called linkage, and it changes the ratio from the normal 9:3 to 3:1 to a 3:1 ratio. With me so far? Okay, so that's perfect linkage. But sometimes, and this is a little bit more complicated, you get something which is in between perfect linkage and not. So let's take you through that. So let's look at three scenarios. This is new stuff. Okay, we didn't do this in lesson. I just made this PowerPoint this morning, so this is kind of taking you a little bit further. Let's look at three scenarios where we have a heterozygous plant, it's not a plant, is it? A heterozygous guinea pig, the F1 generation. And we have both alleles linked on one chromosome. What will the result, if we cross the F1 generation with the F1 generation, what will we get in the F2 generation? Okay, so let's imagine we're looking at guinea pigs here. They've got big B is a dominant allele for brown, small b is a recessive allele for white hair, then we've got big L for long hair and little l for short hair. So let's look at the chromosomes of this guinea pig. So let's say that on this chromosome here, let's say it's chromosome number 1. Okay, so chromosome number 1 carries the two genes, the gene for hair color and the gene for hair length, and they're very close to each other. So this guinea pig here, it's got brown hair and it's got long hair, but it's heterozygous. Okay, so on one chromosome it's got big B, big L. On the other chromosome it's got little b, little l. But the dominant characteristics show up in the phenotype, which is why it's got brown long hair. Now, if that guinea pig bred with the guinea pig with the same genotype, then here's what would happen. We would only be able to pass on one combination, so either a big B, big L, or a little b, little l. So we'd have a genetic cross, which we'd lay out like this, which would give us these results. So if you look at this, these genotypes, you'll work out here that this one, and this one would all have brown long hair, and this one here would have white short hair, and that's what we'd see. So there's the sort of expected genotype expected phenotypes there. So it's 3 to 1, sort of perfect linkage. Right. Well, here's another scenario. In this scenario, we've got the same heterozygous guinea pig breeding with another guinea pig with the same genotype. But in this case, the two loci where the genes are physically located, they're not quite as close. So the big B's up here, the big L's down here, little B's up here, little L's down here. So heterozygous genotype, but now the alleles are a bit further away on the chromosome. So in this case, we're going to need to draw a bigger Punnett square for the cross. So I've tried to sort of show that most of the time, the big B and big L will stay as a pair. Most of the time they'll be inherited sort of together on one chromosome, like this, or like this. But sometimes we will have crossing over occurring. Okay, so crossing over can disrupt this ratio. And crossing over is when these two chromosomes during metaphase of meiosis 1, prophase and metaphase of meiosis 1, there is exchange of genetic material. So maybe this branch of this blue chromosome might exchange with this red chromosome. And in that case, this big B would sort of jump over here and this little b would jump over here. So we call that a recombination event. So most of the time, we'd be looking at the alleles inherited as a pair. So this would be most of the time we'd get this sort of genetic cross going on. But sometimes we'd have these recombinant gametes forming. So for example, this gamete here, big B, little l, how would that actually form? Well, that could only form if the big B here ends up sort of being on the same chromosome as the little l here by crossing over. So here we'd have a different ratio of phenotypes. So we'd have the same phenotypes that we've got here, but we'd also have some new phenotypes. We'd also have white long hair, and we'd also have brown short hair. And the ratio would be not 3 to 1, and it would also not be 9 to 3 to 3 to 1 exactly, because these would still be more frequent. In another scenario, perhaps you see where this is going, we've got the genes separated on the chromosome. The loci are much further apart, almost at the opposite ends of the chromosome. And actually, in this case, crossing over would happen quite a lot. So we would get a ratio much closer to 9 to 3 to 3 to 1. So I've tried to show that here. Again, these would be the more dominant, the more frequent products, but I've tried to show here by sort of darkening the color of the letters here that we'd almost have the same number of recombinant gametes formed as we would have the gametes which are just without any crossing over. So we'd almost get a 9 to 3 to 3 to 1 ratio. So actually we can use genetics to physically map the position of the genes on a chromosome. So if we're getting linkage with a 3 to 1, we can say that the gene loci are very close together on a chromosome. If we're getting linkage which is almost 9 to 3 to 3 to 1, we can say with some confidence that the gene loci are far apart, but they're linked on the same chromosome. Okay, so that, in summary, is linkage. I'll leave you there with some final notes if you'd like to write those down. I hope that's been helpful, and that's it for today. Thanks.